Hello, everyone. We really appreciate you taking the time to join us today. Microbial Insights is delighted to have Dr. Paul Hatzinger with Aptum, who's going to be joining us. Paul is going to be talking to you today about the mystery and magic that happens with cometabolism. So I'm really excited to hear his talk. Many of you already know Paul, but for those who don't, Paul is the Director of Biotechnology Development and Applications Group at Aptum. Paul holds a bachelor's degree from St. Lawrence University and his master's and PhD degrees from Cornell. Co-metabolism of traditional and emerging DoD contaminants has really been an area of interest for Paul throughout his 25-year career in R&D. His research group at Aptum has been really instrumental in improving our understanding of co-metabolic degradation processes, and he's developed field approaches to really utilize these processes and really better understand contaminant remediation. Paul's definitely an expert in this area. He's authored 90 peer-reviewed papers and book chapters, and he's served as the principal investigator on many research grants. So please note that this webinar today is being recorded, and it's going to be available in the resource section of the Microbial Insights website within the next week. You'll automatically be emailed a link if you registered for the webinar, so there's no need to contact us. You're going to get a link to that recording as well as your certificate for continuing education. After the presentation, we'll have a short question and answer session. So please feel free to type your questions in throughout the presentation. If there's a particular slide, please notate what slide that's on. And then at the end, we'll have Paul go through and answer as many of these as possible. So without any further ado, I'll turn it over to you, Paul.
Great, thanks, Dora. Thanks for the nice invite, and thanks to everybody who has taken some time out to attend today to learn a little bit about cometabolism. So what I'm going to cover today, I'm going to do a little bit of background, talk about some history of research in this area, talk a bit about today's sites and site conditions and how this process is particularly applicable to those conditions. I'm going to go through some fallacies and facts about this process, go through some case studies, 2 in situ, 1 ex situ case study, and that'll cover about the first 30 minutes or so. I'm going to talk about natural attenuation, and particularly of chlorinated solvents, and how this process may actually be contributing to that, but we haven't really been looking very well for it. Okay, so what is cometabolism? It's the microbial transformation of an organic compound that doesn't result in cell growth. So cells can't use these compounds as a source of energy, carbon, or inorganic nutrients. So if we look at this figure on the left, this is kind of traditional growth-linked biodegradation. So our contaminant of concern, in this case benzene, is actually the growth substrate. So there's an enzyme system, bacteria break that down, they form biomass, carbon dioxide, and they grow. You get an increase in cell biomass and cell numbers. And in this case, oxygen is the electron acceptor. And most of the processes I'm going to talk about today cometabolically are aerobic. That doesn't mean there aren't some anaerobic cometabolic processes, but most of this is aerobic that I'll be discussing today. Now with cometabolism, you have a primary substrate, and this is often a small ring compound, phenol, benzene, toluene, or an alkane gas like methane or ethane. The cells grow on that. There's usually a primary oxygenase enzyme that transforms that. In this case, say methane to methanol. And then the organisms form biomass and CO2, just like we saw with benzene. The thing that's unusual here is that these enzymes, these oxygenases, often have a broad specificity. So what they can do is they can also break down contaminants of concern. So the cells gain nothing from this part of the reaction. They do all of their growth on the primary substrate, but we have a sort of a fortuitous process in the fact that contaminants get broken down. So let's go through a little bit of history. So this is a table from a review article from 1972, a gentleman named Raymond Horvath. Now, 1972 is a long time ago. Neil Young had just put out Harvest. The game Pong came out in 1972. A bunch of folks were messing around at Watergate. So this is a long time ago, and yet this is a review article. So this process was first discovered in 1959. And basically a paper was published showing that an organism called Pseudomonas methanica, which is now called Methylamonas methanica, it's a methanotroph, could actually oxidize ethane, propane, and butane to form alcohols and fatty acids, although this organism couldn't grow on those substrates. So this was really kind of a mystery of metabolism. Why would organisms do this? And you see over about the next decade, this is just a review of a variety of different papers that showed cometabolic reactions with a whole variety of different compounds. So around 1970, it was also discovered that this can lead to dechlorination reactions. And these are a couple of herbicides. And it turned out that organisms could actually dechlorinate these aerobically and cometabolically. You know, a lot of the early research on biodegradation, of course, was looking at different types of pesticides. So now as we move into the 1980s, there was a really foundational paper by John and Barbara Wilson where essentially they added some natural gas and air to a soil column and saw aerobic degradation of TCE. Now this is really important, right? Because TCE, much like PFAS today, appeared to be largely non-biodegradable before that time. Some other laboratories then found the enzymes responsible for this, or at least one of them, which is soluble methane monooxygenase. So it turns out that some methanotrophs can actually carry out this process while growing on methane. And as we move into the 90s, there was some very successful field demonstrations. In this case, this is some work by Terry Hazen at Savannah River. And Terry and his group added methane through a series of horizontal wells to degrade trichloroethylene, and this worked very well. And then Perry McCarty did some work using phenol and toluene as substrates. So we're sort of moving down this path of developing aerobic treatment technologies for chlorinated solvents like TCE. So, and then what happened? Well, what happened is we started to understand that there are these other organisms that can anaerobically degrade chlorinated solvents. And one of those was isolated, Dihelicocoides ethanogenes, which is now called Dihelicocoides mccartyi. And this basically forms sort of a whole area of research. And a lot of the cometabolic research on chlorinated solvents was now sort of switching over to reductive dehalogenation. And as an industry, we started more practicing anaerobic bioremediation. This is by adding substrates, creating anaerobic conditions in aquifers, and then beyond that, starting to add bioaugmentation cultures anaerobically. And I would say that this process has been highly effective for treating chlorinating solvents, you know, based on the 90s and 2000s and even into the, past decades problem definition. But I'm also going to argue that that problem definition is changing. So if we look at the face of remediation and we go back 20 or 30 years, you know, what we basically had was a lot of chlorinated solvent sites. And we had a lot of chlorinated solvent sites with high concentrations, milligrams per liter, often under anoxic conditions. You know, these parent compounds are things like PCE and TCE and TCA. And as an industry, over the years, we basically developed a lot of technologies to treat these milligram per liter plumes, including bioremediation, in situ chemical oxidation and reduction, permeable reactive barriers, and a whole variety of other treatment approaches that are still used today. However, as we move to today, some things have changed, and a lot of the plumes that we're left with are actually residuals from these old source areas. They're large, dilute chlorinated solvent plumes with microgram per liter concentrations, not milligrams per liter. The geochemistry is microaerophilic to aerobic. You know, we have some of the parent materials and then a lot of daughter products, things like cis-DCE and vinyl chloride that come from reductive, partial reductive dechlorination of TCE and PCE. You know, we also have co-contaminants mingled with these, and 1,4-dioxane is of particular note. The other thing we have are a range of other emerging contaminants that we weren't really considering 30 years ago, that often occur at very low concentrations, but are of concern in aquifers. And what we have to deal with these dilute plumes is largely pump and treat, and no one really wants to build pump and treat systems anymore because they're very, very expensive. They work well to cut off plumes, but you know they tend to have a long half-life and cost a lot of money. So in this ever-evolving face of remediation, I'd argue that cometabolism is the best approach for a lot of our current problems. Again, on the contaminant side, this includes dilute CVOCs, so where you have, you know, micrograms to hundreds of micrograms per liter of chlorinated solvents, along with things like 1,4-dioxane, which is highlighted here because it's a significant issue. 1,2-dibromoethane, it's a leaded fuel additive. TCP, which was a fumigant, NDMA, which is found in liquid rocket fuels. It's also a chlorination byproduct, BCEE, MTBE, the old fuel oxygenate, and mixtures of a lot of these with CVOCs. So a lot of these share characteristics. Apart from the CVOCs, a lot of these other contaminants, they tend to occur at very low environmental concentrations, and often they have lower regulatory requirements, PPT to low ppb levels. They also tend to be water soluble. They don't sorb well to carbon. They have a low Henry's law constant, so they're not easy to strip. So traditional technologies, carbon adsorption, air stripping, don't work particularly well for these contaminants. And some of the fallacies and facts, I was serving on an ITRC committee for 1,4-dioxane, and in part of a discussion, and someone said, well, this cometabolism is really unlikely to be successful at treating contaminants to the concentrations you need. It's sort of a needle in a haystack, right? Because of competitive inhibition. Growth-linked metabolism is much more likely to be successful. And so what this person was saying was, well, if we have an enzyme and we're feeding a primary substrate, which is kind of the remediation approach, as I'll talk about for cometabolic processes, we're feeding propane, these organisms are growing, and your propane sort of comes into the active site of this enzyme. And what that does is it prevents the much lower concentration of your contaminant from being broken down. This is called sort of classical competitive inhibition. But that paradigm is shifting over time. And in particular, what's being found is that competitive inhibition is very organism and compound dependent. So enzyme affinities differ really widely among different organisms and with different contaminants. And I'll also say and show later on in some of the field demos that this is also very easily overcome in the field. To demonstrate this is a figure that I like to show, and this is some work from Josh Sharp about 10 years ago with Lisa Alvarez Cohen. And also at the bottom of each of these slides, where there's a project associated, I'm listing the project number. And if you search online, so you could search CertUp project ER1417, and a lot of this work is funded through the CertUp, ESTCP, or AFCEC programs. You can actually find the final reports and information and papers that sort of go along with this. But basically, in this study, Josh had two organisms that both degrade propane, a Rhodococcus species and M. vaccae job 5, and he set up an experiment where you had increasing initial propane concentrations, and then he looked at the rates of degradation of NDMA with that propane concentration. And what we see is that for M. vaccae job 5, you get a really fast decline in rate. So if you have any significant propane around, you're not going to see a lot of degradation of NDMA by this organism. However, for the Rhodococcus species, you can see that you can have high ppm concentrations of propane and still have very good rates of reduction of NDMA. So this just illustrates the difference in enzyme affinities with cometabolism. Now, the other thing that's important, and these are some actual field data. This was a site in California, and we were doing propane biosparging there to treat NDMA. And essentially, we did some sparge cycles and looked at concentrations of propane in solution in the aquifer, as well as rates of degradation over time. And the first thing you see is that the concentrations we were achieving were under 250 ppb over time. So this is kind of a flux. So you're adding propane, it's being degraded. You're not putting ppm levels typically, or high ppm levels of your primary substrate into the ground. And the other thing you'll notice is that the rates of degradation are very high. So when this process is working well, over an hour, you're degrading the 200 ppb of propane that you've added. So you do this in cycles, but there are long periods of time where there are very low propane concentrations, so you're really not seeing competitive inhibition in terms of degradation of your contaminant of concern. The other thing that's become apparent is a lot of these organisms have multiple oxygenases, and more than one of these oxygenases can be induced and active at any given time. And a really nice example of this is Mycobacterium tubense, and this is some work by Nick Coleman in Australia. And basically, what he showed is that this organism has at least 6 distinct monooxygenases. ethene and propene, propane and butane, alk B, cytochrome P450. It grows on alkenes, it grows on alkanes, and it degrades a whole variety of different contaminants. So the paradigm here is that with some of these organisms, you can have different oxygenases induced, and it's possible that one oxygenase they're using to grow on the primary substrate, but a whole separate oxygenase is breaking down the contaminant of concern. And a nice example of that, this is some work we did in our lab where essentially we grew this organism on ethene and we added cis-DCE, and in the presence of ethene, the organism did not degrade cis-DCE. In the absence of ethene, when the organism was induced, it degraded it very readily. So this is kind of classic competitive inhibition. However, with 1,4-dioxane, what we see is that in the presence or absence of ethene, the organism degraded 1,4-dioxane. So one possibility for this is that it's using two separate enzymes. So the ethene, it's using the ethene to grow, it's degrading the ethene, but a whole separate oxygenase is potentially breaking down the 1,4-dioxane. So you aren't going to see a lot of competitive inhibition in that case. So the other thing I often hear is, well, growth-linked metabolism is much more likely to be successful as a field application, right? And I think when you have ppm concentrations of things that are biodegradable, that organisms can grow on, that's true. If you can grow the organism on a contaminant, you have very high concentrations, that's a nice approach. But for a lot of these contaminants we're talking about in this sort of dilute plume dilemma, it's very difficult to maintain cell growth on ppb concentrations of a contaminant. If we look at 1,4-dioxane, 92% of Air Force sites have less than 120 ppb. It's hard for organisms to grow and increase in density with those low concentrations. So you can't maintain the high biomass. And the other thing is these organisms that carry out growth link processes may or may not be present in an aquifer. Now with cometabolism, if you're practicing this in the field, you're supplying the primary substrate. You're adding propane or you're adding methane to the aquifer. So you're controlling those growth kinetics. You can achieve high biomass by doing that. The second thing is you can achieve very low contaminant concentrations, and I'll show you a couple of examples of this, but here's a lab study with N-nitrosodimethylamine. This is a Rhodococcus species, and this is 8,000 parts per trillion or 8 ppb, and this organism growing on propane can degrade that down to less than 2 parts per trillion, so less than 2 nanograms per liter in the presence of propane. The other thing is these organisms tend to be pretty widely distributed. But of course, cometabolism is very hard to apply in the field, right? You may have to add a gas, some of those gases are flammable. And the answer to that is also no. Good engineers can very easily design systems to handle flammable gases. Many of us have propane, grills in our backyard. We have gas heat in our houses. It's not very hard to design systems to be able to handle flammable gases. But how do you add those to an aquifer? Well, that must be very difficult. And the answer there is also that it's not. And there are many different ways to do this. First way is biosparging, where you basically just sparge the gas, usually in air, into an aquifer. This is kind of based on air sparging, where this technology used to be used to strip volatile contaminants like BTEX from aquifers, but you would have very, very high flow rates. These are very low flow rates where you're just trying to apply a gas to the aquifer. It's flexible. You don't need a pumping infrastructure. You generally have a pretty good radius of influence. It works well under confined or unconfined conditions. One of the issues is preferential flow paths, so you certainly need to do some testing there. But overall, this is a very good approach to get your substrates into groundwater. Second is groundwater recirculation. So in this case, you remove your groundwater, you actually add your oxygen and inorganic nutrients and your cometabolic gas, and then you re-add it to the aquifer. So you create a bioactive zone within the aquifer. This is really flexible. You can get very good amendment distribution, and you can add liquids and cultures. If you have to bioaugment, that makes it very easy. If you need to add soluble nutrients or other amendments, you can do that. You do have to have a pumping infrastructure, and biofouling can be an issue. Third approach are passive in-well diffusion devices. There's something called a Waterloo emitter. Essentially, you put gas pressure on tubing, and the gas just diffuses through that tubing into an aquifer. The nice thing about this is it's very simple. The downside, it tends to have a very poor radius of influence. You need a lot of wells in order to be able to do this. A few field considerations. First is inorganic nutrients. If you are supplying a lot of carbon to an aquifer, which tend to be sort of a somewhat oligotrophic setting, you're often going to run into a nutrient deficiency. This may not happen if there's a lot of nitrate in the aquifer, if it's aerobic to begin with, and you already have sort of natural nitrate there. But if you don't, you're often going to have to add some type of inorganic nutrients, and nitrogen in particular. And here's a nice example. This was a study with dibromoethane. I'll show a little more of this, but this was a site on Cape Cod, and essentially we ran some microcosms, and if we added just ethane or propane, we saw no degradation of this contaminant, EDB. If we added just nutrients, we saw no degradation of the contaminant. However, when we added the gases, ethane or propane, plus the nutrients, that's diammonium phosphate, we saw very good rates of degradation. And in the case of ethane, the contaminant was degraded to less than 15 parts per trillion. So I'm going to show you a case study in a second, and this was some of the microcosm work that sort of led up to that and led up to us using ethane and diammonium phosphate in the aquifer. But in the absence of adding nutrients, this process would not have worked. The other thing is, all gases aren't created equal. The amount of gas that you use, if you're using, ethane or propane, isobutane to add to an aquifer, is fairly trivial in terms of the cost of these projects. So, using a gas that's of lower quality because it's cheaper is really not worth the potential headaches. And if we look at propane that you get from one of the box stores, it tends to have up to 10% of propylene, which can be problematic, as well as mercaptans. And this is one of an organism that we use frequently called our Ruber ENB425. It grows great on 99% pure propane. It doesn't grow well in the consumer grade propane. There's inhibitory factors there. So, it's important to use the pure grade materials. Now, case studies. I'm going to show 3 very short case studies. The first one will be looking at EDB in groundwater in a research system. The second one is going to be 1,4-dioxane in dilute CVOCs using propane biosparging. And the third one will be ex situ treatment of NDMA in a propane-fed bioreactor. So there is also potential applications of cometabolism for, ex situ treatment. So let's start with dibromoethane or EDB. Now this compound, simple alkane, was widely used as a lead scavenger in leaded fuels. It's still used in avgas. It's a potent carcinogen, and it has a 50 part per trillion US federal MCL. It was phased out along with leaded fuels in the 1980s, but it persists potentially at thousands of gasoline and avgas sites, and again, significantly above its MCL. When you have a 50 part per trillion MCL, concentrations of tens to hundreds of ppb are significantly problematic. The good news with EDB is it does hydrolyze slowly over time, so concentrations are presumably decreasing, but it's a very widespread issue at former fuel spill sites. And it has a lot of these characteristics I talked about at the beginning. It's soluble, has a relatively low KOW or Henry's law constant, so it's somewhat hard to strip from water. It doesn't sorb particularly well to carbon. There are some organisms that degrade it aerobically or anaerobically, but again, concentrations are frequently very low in an aquifer. So whether that's going to be effective, remains to be seen, but it's likely that these growth link processes aren't going to be very effective. But there are a variety of different substrates that can promote its co-metabolism, including ethane, propane, methane, and ammonium. So this project was conducted with funding through AFCEC, and this was at a EDB plume at Cape Cod. So the fuel hydrocarbons had long ago been cleaned up, but there's a very long EDB plume, and this is one of several at Joint Base. At Joint Base Cape Cod, which used to be called Mass Military Reservation or Otis Air Force Base. It's currently being treated under a pump and treat scenario. And essentially, after some field work, we designed and installed a recirculation system, basically just to test this cometabolic approach in the aquifer. And if we look at this graphic, we used an existing extraction well from the system. Water was removed, basically with EDB. It's amended with ethane, oxygen, and diammonium phosphate. And I showed you some of that treatability work previously rather. That's the reason that we use these amendments. It's then re-injected into an injection well. And then you sort of create a bioactive zone. So what we end up having is a big recirculation loop here. We continually amended with ethane gas at about 1 to 2 ppm. We periodically added inorganic nutrients, and oxygen was added separately through cylinders, but everything was sort of mixed up in this Connex box and added and recirculated within the aquifer. So what the results show, and in the left, these are ethane concentrations. So, we mixed for about 90 days to sort of equilibrate concentrations of EDB in our treatment zone. Then we started adding ethane and oxygen, and we see ethane start to spike in our monitoring wells. And these are data from that whole series of monitoring wells that had been installed in the recirculation plot. We see concentrations up to about 12 or 1300 ppb. But now realize we're continually adding ethane throughout this whole process. So essentially we're adding ethane at kind of this level, but you see within the treatment plot, those concentrations fall off and they get to the point here where you're not detecting ethane in other than the injection well. I didn't plot those data, but injection well data were up here at the 1 ppm concentration. But what's happening is the organisms are so rapidly degrading the ethane that you're not seeing detectable levels even 5 or 10 feet down gradient of your injection well. Now, if we look at the data, and so the concentrations of EDB in this plume are 0.3 ppb, or 300 parts per trillion on average, but the Massachusetts MCL is 20. So these are still 15-fold higher than the Massachusetts MCL, so they need to be treated. Here's our sort of mixing time. We add ethane and oxygen and then inorganic nutrients, and we see in all of these wells that are well connected, we see a significant decline in EDB concentrations. Now this is our recirculation water. So this is coming in, this is being pulled from the plume itself. And the thing that was interesting is we were also having an impact on basically on the plume influence. So enough of our treated water was mixing back in with that system that was declining with time. But importantly, by about day 200, when we shut the system down, we had basically reached not only the federal MCL of 50 parts per trillion, but the Massachusetts MCL of 20 parts per trillion in each of our monitoring wells that were connected to the system within the plot. The other thing that's kind of interesting here is that we see when we shut everything down, there's not a lot of rebound in a number of these wells. There's a few that are starting to rebound after about 100 days. So I think what happens is you've built a lot of biomass, and I think these organisms are continuing to biodegrade the contaminant just by using the biomass in the system. So this was a very highly effective approach for EDB in this aquifer. Now, the second demo I'm going to discuss is for 1,4-dioxane. This is a well-known stabilizer in trichloroethane. It's found at a fair number of chlorinated solvent sites. Again, very soluble, low KOW, low Henry's law, so it's hard to treat by stripping or sorption. There is growth-linked metabolism for this compound. There's a famous organism called CB1190 and several others. There's nothing that degrades it anaerobically, but there are a whole variety of different organisms that can also co-metabolize it, growing on a variety of different substrates. Now, this site is Vandenberg Air Force Base in California. It's a former tank maintenance site, had 1,4-D at pretty high concentrations, about 100 ppb to a ppm, as well as low-level VOCs. And the goal of this was to demonstrate in situ 1,4-D treatment and also look at potential treatment of the different volatile organic compounds that were there as well. So this was a field pilot. This aquifer has a shallow, an intermediate, and a deep water-bearing zone. The formations here are relatively tight, so it's fairly hard to sparge gases here. You could probably do a recirc type design or just sparge slowly, but for this demo, kind of a limited scale demo, basically we worked in the deep water-bearing zone, had one sparge well, and then a series of monitoring wells. And this was a well that had been installed previously, 5B, so it was kind of broadly screened across different layers. These other wells were put in for the demonstration, and this well in the upper zone was used as a control. So basically what you see is, propane gas and nutrients were sort of added to this well, and then monitoring was done at all of these different wells over time. And I should say that this work was led by Dave Lippincott at Aptum. So the system used here was a couple of trailers, and essentially one trailer has a compressor. It compresses air, it feeds that air to a second trailer, which then feeds in the appropriate amount of your alkane gas. So you have one trailer with a compressor, one that's explosion proof that feeds in the gas. You see the gas cylinders on the outside of the trailer here. This started with just some air sparging to look if there was any loss of contaminants before you started adding your propane gas. Propane addition was started. This was bio-augmented. There were certainly some propanotrophs in this formation, but it took a lot of time. So this was kind of an insurance policy, adding this 425 organism. And you can see, I talked previously about propane usage. Well, it's less than a pound a day. So basically you're adding at about 40% of the LEL. This is done in cycles. And with these types of systems, they're frequently automated and you can control them from outside. So, you can go and monitor your propane concentrations, and then you can add accordingly so that, you're pulsing in enough propane to keep the organisms alive and active, but not so much that you're creating a lot of competitive inhibition. And there were also some periodic nutrient injections. So basically, if we take a look at the data here, so the sparge well had fairly low concentrations. We see a pretty rapid decline, and this was sort of the beginning of air sparge only. We don't see a lot of declines during that phase, some increases. This was basically when the propane sparging started and then the bioaugmentation. But for all of these wells that were connected, 34B, 47B, 2B, we see very good declines in 1,4-dioxane after treatment starts. No changes in the 5A well, which is up here. This was the control well. And then sort of odd data from the 5B well, but again, this was a previously installed well that screened over multiple layers, and there may very well be a confining layer here. So it may have only been a small amount of water that was coming into this well, and this well didn't see an awful lot of propane. So that makes a good deal of sense. But anyway, this worked extremely well. And you can see that basically in each of these wells got greater than 99% removal down to the 1 ppb range. This 34B well, a little bit less, but 95% removal. And then these wells that weren't impacted, you had very little removal. Now, as far as I know, the current contractor at this site is actually using this process full scale. So they're treating this entire plume. This was, again, just a pilot study, but they're treating this entire plume using this process. Now, the other thing that was interesting here is there were low concentrations of some chlorinated solvents, 1,1-TCA, 1,1-DCE, and you probably can't see this easily, but this is 50 ppb, this is 300 ppb, and then a little bit of TCE at about 12 to 14 ppb, with none up in that top formation. But again, if we look at all of these wells that were well connected to the system, we get non-detect levels of TCA, non-detect levels of DCE, non-detect levels of TCE. With, this was the well that wasn't as well connected. You don't see treatment as much efficacy there, although there are some declines. So, and the other thing that that was done here is that microcosms were set up just to ensure that this was biodegradation and not some sort of stripping that had been going on in the aquifer. And basically groundwater only was taken from each of these different wells. It was amended with propane and nutrients, a killing agent, so sodium azide or nothing. And for each of the different wells, those that received propane and nutrients, there was complete degradation of the chlorinated solvents. There were no losses in the killed samples or no losses in the samples that just had just oxygen alone. So that confirmed that a vast majority of this, in fact, was biodegradation. So joint treatment of 1,4-dioxane and chlorinated solvents. So my last case study here is treatment of NDMA. NDMA is a carcinogen. It was in liquid rocket fuels as a contaminant. It's also a chlorination byproduct. This is why you shouldn't eat a lot of hot dogs. You can form this in your stomach. It's also most recently been seen as a drug contaminant, in Zantac. So there's a lot of interest in NDMA. Has all those same characteristics. There's never been an organism isolated that can grow on this, which is pretty interesting. But there's a lot that can co-metabolically degrade it. And this project was conducted at the White Sands Test Facility in New Mexico. This was a plume that contained NDMA with CFCs and VOCs. And the current treatment method is air stripping with UV treatment. So it's kind of a commingled plume here of all of these different things from some former liquid rocket fuel testing. So the goal of this project, which was an ESTCP effort, was to see if you could treat the NDMA instead of using UV, which is expensive, with a bioreactor and a co-metabolic bioreactor. So this is a photo of the system. This is about a foot in diameter, 12 feet tall. This is filled with granular activated carbon. This is some nutrient tanks and a control panel. And essentially, so water is fed into this plant. It goes through an air stripper, and then a slipstream was taken and run through this fluidized bed reactor. It was fed inorganic nutrients, which were urea and diammonium phosphate. And then it's amended with propane and oxygen. And so that amended water with the contaminant goes into this fluid bed reactor. It's filled with granular activated carbon, and biomass grows on that carbon. And then you get treated water coming out. And there's a recycle component of these fluid beds that allow you to change the residence time in the bed. So you can either have one pass and a very short residence time, or a lot of water that recycles, which gives you a much longer residence time within the bed. And these are just some photos I took many years ago. This was a sand-based fluid bed, so it had sand particles, but you can see, the thick biomass that grows up on these particles. It's just a nicer photo with the sand than the GAC. You don't see very much, but you get a huge amount of biomass in these reactors, which makes them very effective. And this was one of the first that was a co-metabolic design system. And I'll mention that, Todd Webster and Virogen designed and built this. And here are some of the data, and what we're looking at across the top here are basically decreasing residence time in the reactor from 60 minutes to 10 minutes. This is the influent of NDMA coming into the reactor, and again, it's only coming in at a part per billion. Before we inoculated and started adding propane, you see influent equals effluent concentrations. We add the organism, we start adding propane, and you see declines in NDMA. And we sort of get to the point, and we're also decreasing residence time, but at either a 20 or 30-minute residence time, our effluent concentrations were between 3 and 3.5 parts per trillion. So this is, this is orders of magnitude reduction. This met the site needs, which was originally 10 parts per trillion in their effluent, but was lowered to 4.5 or 4.4 parts per trillion at some point. But this is a commercially, viable system at these HRTs. And then we reduced it further to 10 minutes. And you can see it starts to fall back. So you're probably reaching that point where, this is too rapid a feed rate. But then we also ran some, studies turning the system on and off and seeing how it recovered, et cetera. But essentially, this showed that in a fluid bed, you can get very good treatment of this contaminant, even at pretty short hydraulic residence times using co-metabolism. The other thing that was interesting here, we looked at propane monooxygenase. So this was just some work at MI, and this is a few years back now, but so it just showed that we had really, really high levels, 10 to the 9th, cells per gram of carbon within the reactor. We also looked at ammonia monooxygenase, which was at much lower concentrations, thinking that maybe we were feeding ammonia as a nutrient source. Maybe it was also acting as a source of a co-metabolic substrate. But the other thing that was really interesting is the organism we added, Rhodococcus rubra ENV425, didn't show up when we actually did later analysis. It was basically mycobacteria species. So probably what happened within the reactor is that organism was outcompeted by native organisms that had the same metabolic capabilities. And that's kind of what you find with some of these co-metabolic processes. There's lots of organisms. They all tend to fall under the same order, which is actinomycete type organisms, which include nocardiiforms and mycobacteria, Rhodococcus species. And they tend to be the ones that have these capabilities. So sort of that's it with the active part, and I'm just going to finish up my talk a little bit sort of discussing aerobic co-metabolism and whether we're missing a significant component of natural attenuation of CVOCs. So when we think about monitored natural attenuation of chlorinated solvents, this makes the most sense as a long-term remediation alternative for many different sites. What we have are a lot of sites where we have low concentrations over extremely large areas. So a lot of in situ approaches are just not going to be cost effective. The areas are too large, to add carbon sources or to add organisms. So natural attenuation, for those of you who are unfamiliar, that basically means that what you do is you prove that you either have a stable plume or a shrinking plume, and there's a whole series of factors that we understand for anaerobic processes where you can then say, okay, this site is subject to MNA, so we know that this plume is not going to expand, it's going to shrink, and over a period of time, it's going to either go away or not cause further problems. And over the past 25 years, most of our M&A focus for chlorinated solvents is really on reductive dehalogenation, but we really don't know what to look for when we have bulk aerobic sites. And I think most practitioners don't believe there's any meaningful degradation occurring for many solvents, things like TCE, when you have bulk aerobic conditions. And this was something I came across, which I really, really like. And this was Perry McCarty, 1997. So, Perry McCarty is one of the great forefathers of bioremediation at Stanford. And he was giving a talk and basically said, there's ample evidence of reductive transformation processes of solvents and their products. So we understand that this is occurring at sites where conditions are reducing. But he also said aerobic metabolism of TCE would only occur if there was sufficient dissolved oxygen, a suitable electronic donor, and sometimes odd things like methane, ammonia, or phenol. And basically said such circumstances are not likely to occur. Therefore, co-metabolism of TCE is probably of little significance in terms of natural attenuation of TCE. And I think even today, most people would probably agree with that. And I'm going to try and convince you that that may in fact not be true. So when we look at reductive dehalogenation here on the left, we understand now pretty well the organisms that do this. We understand the substrates. We have Dehalicocoides. There are daughter products that we can readily measure. So we know what geochemistry this occurs in. We understand the organisms. We can understand and detect a lot of the genes. We know what electron donors are necessary. We can measure the daughter products. We can use CSIA to sort of prove this process. So, dating back to the late 90s, you could start building cases for natural attenuation. You basically just have to show the conditions, the organisms are there, the genes are there. You have conditions where these contaminants are degrading. We understand this pretty well now. Now, with aerobic co-metabolism of chlorinated solvents, we need aerobic aquifers. Is there some minimum concentration? Do we know the key organisms? How about the genes? Substrates and inducers, are they present? Are they likely not present? Are there any unique degradation products? Can we use CSA? Often we can't because the fractionation is relatively low. So can we build a case for MNA for aerobic co-metabolism? And what I'm going to kind of show you is that we can check a number of these boxes now, and some of these results might be a little bit surprising. And not CSA, but there's another technique that's now come up where we can actually document this process occurring in aquifers under aerobic conditions. And I think that's really exciting. So aerobic co-metabolism. So what Perry said is, well, you're going to need toluene or ammonia or something as an inducer or a substrate, and these are unlikely to be found in aquifers. If we go back a bit, some work by Rob Steffen a number of years ago, but basically what he showed is that the chlorinated solvents themselves can induce key oxygenases at times. And in this paper, he showed that basically TCE and other contaminants could induce toluene dioxygenases and monooxygenases, phenyl hydrolase, alkene monooxygenase. So you don't need the specific substrate of concern to be an inducer. The contaminant itself can do that. The other thing is the primary substrate is not necessarily the only growth substrate for these organisms. So you need, something for induction, something for growth. And this is a great paper where these scientists were looking at root exudates that come from plants. and they developed some enrichment cultures from sort of the rhizosphere of plants. And these are the types of compounds that you find, these secondary plant metabolites that get released into soils. And they were looking in soils. And essentially what they found is that each of these different metabolites promoted aerobic cometabolism of cis-DCE. So this is not toluene, it's not phenol, it's not sort of the primary growth substrate, but each of these different naturally occurring substrates could promote that process. Okay, so what else? What other substrates and inducers might be out there? Well, of course, we know anaerobically our chlorinated solvents degrade to ethene, ethane, and methane, which then can move further in an aquifer to aerobic conditions. So those can be inducers and substrates. What about fuel spills? Well, what happens at fuel spills, particularly if they're sort of mixed, is you tend to get oxidation of the small molecules, things like naphthalene and cyclohexane, and you're forming these compounds that can almost certainly be inducers. How about carbon release from cell death and lysis when we grow a lot of biomass? I think at that EDB site at Joint Base Cape Cod, that's what we were seeing is continued co-metabolism without adding ethane gas due to these products. How about carbon from past remediation activities? You know, one of the great things I heard Perry McCarty say at least 15 years ago, he said, you know, someday I think what we're going to be doing is cleaning up emulsified oil spills, because you know, a lot of practitioners are adding these huge amounts of carbon to aquifers to treat chlorinated solvents, you know, large amounts of emulsified oil and other things, and realize that, you know, those also get oxidized over time. And so you can be getting a lot of daughter products released from those. I mentioned root exudates previously. You know, if you have areas with landfill leachate, there's all sorts of carbon in there. You have a lot of natural humic and fulvic acids that occur. And you can see these are ring compounds that can get broken down. And if you take a look at some, this is a really, again, an old USGS report where they just looked at dissolved organic carbon in aquifers, you know, the mean concentration was about a ppm. So if we're talking about natural attenuation, you know, if organisms can slowly break down some of this carbon, then this could be a source of substrate over time. Now, how about those co-metabolic genes? You know, no one really spent a lot of time looking for these. Everyone was always looking for, you know, your reductive genes, your genes that are known to, you know, for reductive dehalogenation. But it turns out, so Microbial Insights has a database, and they have basically a quant array analysis that is most frequently done for chlorinated solvent reductive dehalogenation, but they include a lot of the aerobic targets in that assay. So if you look at their database, what you find out is that these are all aerobic cometabolic genes. These are toluene monooxygenases, toluene dioxygenase, methane monooxygenase, phenyl hydrolase. So in all of these water samples, these are occurring at about 50 to 80%. And I would bet you that a lot of these are anoxic water samples. And while these concentrations aren't high, they're high enough, you know, you're at, you know, 10 to the 6 per liter in this case, that these could have some significant activity. So these organisms are pretty widespread. And this is just an example of one of these quant array printouts, and this is from a biotrap at a site. And you see, you know, you're looking for dihalococoides, TCE reductase. This is very commonly done today. And you see pretty good concentrations of some of these. You see Dehalobacter at 10 to the 5th or 10 to the 6th per bead. But if you look at the second part of this, which no one ever looks at because they're mostly worried about these reductive processes, here's the aerobic cometabolic genes. Well, look at this. We've got phenyl hydrolase here. We've got toluene oxygenases. Now, these are aerobic organisms. These are aerobic, you know, genes that occur in aerobic organisms occurring with strict anaerobes. So these are occurring on the same beads in the same environment. Okay, so what's going on here? Well, you know, this is kind of supported by some recent research, and this was a nice paper by Tim Mattis, and he went out and looked at water samples, and he looked for dihalococoides, and he was looking for, basically, he's interested in cis-DCE and vinyl chloride oxidation, and these aerobic ethene oxidizing organisms, and he found that they were together in 99% of water samples and expressed together, so they were actually expressed in 60%. So again, you have aerobic and anaerobic organisms together. And some of Tim's more recent work, he did some coring, and actually found the same thing. So they're essentially were co-located in cryocores. So these were aerobic and anaerobic organisms that degrade vinyl chloride occurring together. So, I think what this is pointing to is we all have this concept, right? And this is kind of drummed into your head. It's sort of a means to so that you can try and understand electron acceptors and respiratory processes. But you always see a drum and there's a big spill, and then what you have is that sort of spill moves along. You consume all of your oxygen, you get into nitrate reducing and then iron reducing conditions and sulfate reducing, and then eventually end up in strict anaerobic conditions with methanogenesis. So what would you expect to find? Well, you'd expect to find aerobes out here and nitrate reducers and iron reducers and sulfate reducers, and it's just sort of this paradigm that we've all sort of grown up with, but I don't think it's even remotely realistic. I think what we really have is a world of niches in these environments, and we have niches here as well as we have out here, where you're probably going to find a wide variety of organisms. So just a little more on that concept. So, here's kind of one example. So we know one of the problems with these dilute plumes is these chlorinated solvents have become entrapped in clays and other things, sort of fine materials, and they're slowly leaching out. So you could have environments in an aquifer, let's say that's mildly aerobic. Maybe you have a half a ppm of dissolved oxygen in this aquifer. And you've got TCE trapped, and you could be forming some methane in here. It can be very reducing because you don't get oxygen diffusing well into this environment. But as that methane and TCE come out where you have some levels of oxygen, you may be getting TCE oxidation. So perhaps you're getting reduction in this environment at some level and then oxidation here. But then you would say, well, as Perry said, you're going to need oxygen. So if you only have a half a ppm of oxygen, this probably isn't going to occur, right? Well, I would say that's very much wrong. This was a nice paper by Jim Gossett about 10 years ago. And what he did is he was trying to understand vinyl chloride losses in anaerobic environments. So he set up a system where he could add extremely low concentrations of oxygen, 0.1 ppm, 0.02 to 0.1 ppm of oxygen into these little bottles continuously. And what he found was that aerobic organisms would grow with that little oxygen flux to degrade vinyl chloride. And this occurred for both pure cultures and in microcosm studies. So you don't need a lot of oxygen for these processes to be occurring. The main problem that we have with this is trying to have daughter products. It's very hard to document degradation aerobically of something like TCE because the products you get are things you can't measure by GC purge and trap. And they ultimately degrade to CO2. So it's really hard to see cometabolic activity. There is no vinyl chloride, there's no cis-DCE daughter product or ethene that you can easily detect. And that's been kind of hard to overcome. And that's also part of the reason we, if there was a daughter product that you could easily see, I think we could tell that this was going on. But there isn't. Now, what's changed in the last several years, and this is a CERTUP project, is that an assay was developed down at Clemson, and this is some work with Todd Wiedemeyer, who unfortunately has passed away, and John Wilson, David Friedman, and grad students. And what they did is they developed an assay using ultra-pure radio-labeled TCE. They collected groundwater samples from a bunch of different sites, and then they looked for cometabolic activity. So they looked for aerobic oxidation of TCE at these different sites. Now, in this field, we've used C14 for a long time just to try and understand the fate of different compounds. But what's different here, these compounds always have some contaminants that cause issues, a couple percent. So what David Friedman's group did is they actually ran this radio-labeled TCE through a GC and injected it directly into bottles, directly into microcosms. And that gives you basically a super sensitivity to look for any transformation of the compound that you can't see by GC. You can see 0.001% transformation of 14C TCE if you add enough of the product, and they add about 1 million DPM. Essentially then remove samples over time and look at the daughter products. This is run against controls because you can get some autolysis of the TCE. So basically you look at the amount of in your samples, the amount of C14, and you can convert that to the amount of TCE. And then as it gets oxidized, you get these soluble products. So you can collect samples. So those soluble products include carbon dioxide, and then, as I showed previously, some of these other soluble compounds that you can't strip. So if you take that sample, you raise the pH, that'll maintain the carbon dioxide as carbonate. If you sparge the sample, you remove all the parent TCE. So all you have left are soluble products. In this case, the carbonate, and then whatever those daughter products were from the TCE, which I showed previously, glycolic acid and other things. You can then precipitate out the carbonate with barium, and that'll give you just the soluble daughter products. But the most important thing here is you can use this assay to very sensitively look at aerobic oxidation of TCE. And this is just one example from one of the sites. And what we see here is daughter products, soluble daughter products evolving. And from that, you can determine a half-life. So in this water sample, basically you had a rate of aerobic degradation of TCE that amounted to a half-life of about 12 years. And if you look at all of the samples they looked at, 3 of the 5 sites showed some clear cometabolic activity with half-lives of about 0.3 to 107 years. And this is just with groundwater. So this is showing cometabolic oxidation of TCE in the field. And this assay is commercially available now. So, I would hope people would start to think about this a little bit, because I think it's really important. This was a site we had done some work with David Friedman, and this is a site. It's a mulch barrier that was installed to treat TCE at a site in Maryland. It's a Superfund site, and we went and took some samples right from the middle of that barrier. This is a TCE plume. This is the barrier. And David did that work with one of his grad students. This was part of his master's project. And essentially, if you just took soil and groundwater from this barrier, there was aerobic degradation with a half-life of about 2.4 years for TCE. If you added some methane or methane and nutrients, that went to a half-life of less than a year. So this barrier is designed for anaerobic treatment, but clearly there's aerobic treatment going on there as well, or there's a tremendous potential for aerobic treatment of TCE. Now, I'm just going to finish up with a couple slides on 1,4-dioxane, and this is some really good work Dave Adamson did with Chuck Newell and Shaylee Mahendra, Hunter Anderson, and they basically looked at the Air Force data set, and by looking at sort of plumes and concentrations of 1,4-D over time, they determined that 1,4-dioxane was degrading in a lot of these plumes with half-lives that ran from less than 10 months to several years. And there was a correlation with oxygen. So you had higher rates when there was oxygen present, lower rates with metals and CVOCs. And what this suggests is that this is a biological type process. It could be growth linked, it could be co-metabolic. So now, as part of an ESTCP project, basically the C14 assay is being converted over now. This is very new work. Again, David was kind enough to send me a little bit of preliminary data, which will be published very soon. But essentially, he's basically developed the same assay with his research team. And now they've taken samples from 10 sites. And there were statistically significant rates from 9 of those 10 sites for aerobic 1,4-dioxane degradation. Now, those half-lives in general were fairly long. But again, this shows that there is an assay to do this. There's a way to measure 1,4-dioxane degradation using C14 label. And the other thing that's really interesting about this is when you have these long half-lives, you can also use this assay to look at limitations. What if you add a co-metabolic substrate? Is there toxicity? You can determine that. So this was just some data sort of vetting the assay with a pure culture, but it just shows that this is available. And this is really interesting work coming up. So let me just finish with a few final thoughts here, and I'll wrap up. Co-metabolism may play a really important role in the attenuation of VOC plumes. New tools are becoming available to quantify secondary lines of evidence. qPCR of the genes, gene probes, which I didn't talk about today, and I think the C14 assay is really important. And I think we're getting to the point where you may be able to start building a case for aerobic co-metabolism and natural attenuation. So kind of a different mindset or paradigm. And to summarize the whole talk, what I'd like to get across is that this new research shows the wide versatility of these organisms. They're widely distributed. They use many substrates. This can be a highly effective approach for a lot of different contaminants. For those of you who are practitioners, consider starting to use this. You can achieve very low concentrations. And a lot of times growth link processes are not going to be very effective. And, these are just a few recent papers showing, cometabolic degradation of plastics in landfills, or aerobic, cometabolic degradation of insensitive munitions, and also treatment at acidic conditions, which it's very hard to do with CVOCs when you, try to do that reductively. I'd like to acknowledge a bunch of folks, who funded this work. CERTIP ESTCP and AFCEC funded a lot of this work, and you can get the reports and papers, and then a lot of different people here who contributed slides or discussions or were PMs on these different demonstrations that I showed. So thank you to everybody. And I'll wrap it up there and answer questions if there are any. Thank you very much, everybody.
All right, thank you so much, Paul. That was an excellent presentation. We do have a few questions here, and so for those of you that want to hang around for a few minutes longer, we'll try to answer as many of these as we can. So Paul, the first question, I think it's a good one that a lot of people have, is when you're talking about adding this lab analysis at a site, how much of your budget has to go to the lab analysis portion? Is that comparable, or does it even out to if you just did like a pump and treat? You're trying to do all this bioremediation. Is the lab analysis and the study of this as costly as an active treatment system?
Oh God, no. No, I mean, if you talk to any military RPMs, the last thing they want to do is put a pump and treat system in, because they become eternal. So you have not only the capital cost, you have the O&M over what ends up being decades often, because what happens is these plumes, you reach a point where you're treating very little contaminant, because it's whatever's, particularly for these dilute plumes, it's what's leaching out of remaining reservoirs, clays, tight formations, and things like that. So that can end up being millions and millions of dollars. Whereas these types of analyses, and even if you tried to put a package together, say to try and document natural attenuation, you can do the C14 assay relatively inexpensively. So the gene probes, are relatively inexpensive, as so basically you're talking about, with all your sampling and everything, I hate to throw numbers out there, but you're in the tens of thousands of dollars versus just putting in a pump and treat system with the millions of dollars. I mean, maybe at the end you have to end up there, but that's not. It's worth doing the lab analyses to try and see if you can document a natural attenuation. And the other thing to think about, which is proving to be successful, is even when you have some of these dilute plumes, if they're not that wide or that deep, you can put in sort of trench sparge barriers, or just horizontal sparge wells as a cutoff barrier, and add cometabolic gases, so that you can do an active treatment and potentially also prevent a pump and treat system. And often these systems can be off the grid, and Dave Lippincott is just overseen a very successful ESTCP, where essentially they put in a cutoff barrier for a vinyl chloride cis DCE plume down in South Carolina at Myrtle Beach for an old Air Force site, and that worked very, very well to cut this plume off at a relatively low cost.
Great. The next question is, can you talk about the ratio of propane to DAP that is needed for EDB cometabolism?
Yeah, so it's the old rule of thumb. Again, if you go back to some of like Rittman and McCarty and some of those engineering sort of books, on a molar basis, my recollection is it was always like 100 to 10 to 1. So you don't need, you need a percentage of nit, and it's mostly nitrogen. It's not phosphorus, it's typically nitrogen. But there's a couple of ways that you can do that. If you have a research system, in that Joint Base Cape Cod project, a couple of times a week, we just had an automated system where we dosed a little bit of ammonium, diammonium phosphate. It ends up being just a few ppm in the water, because you don't want to add a whole ton of ammonia to an aquifer either. And we never actually saw any ammonia, and we never saw sort of any significant nitrate coming, because you can get nitrification of that as well. So it's relatively small amounts. And on the gaseous side, what we've started doing is actually using ammonia gas and adding small amounts of ammonia gas to an aquifer, but it's a very small percentage compared to what you're going to be adding is, say, your propane, which is also not very much. As I showed, for some of the systems that I showed in the demonstration, you're adding less than a pound a day. I mean, you're not using much propane to begin with, so it's going to be much, much less in terms of your inorganic nutrients.
Okay, on sites where you see that the hydrocarbons are depleted, but EDB is left, do you ever see 1,2-DCA present?
Yeah. I mean, so those were used. Both 1,2-DCA and EDB were used as lead scavengers, and their microbiology in terms of organisms that degrade those cometabolically and reductively are pretty similar. I'm trying to think of examples, but there are certainly sites out there that have 1,2-DCA from from leaded fuels. I'm more familiar with the EDB again, and I think part of that is because of some work Ron Falta did many years ago, where he basically documented how much EDB is present in some of these sites, and also the fact that it has such a low MCL. So when people look for EDB, they look for it down to very low concentrations. So yeah, I mean, there are going to be some sites that also have 1,2-DCA, and it's just going to depend upon which lead scavenger was used in those different fuels.
So in the first case study that you showed, what factors led to selecting ethane as the substrate?
Yeah, so part of that was the fact that when we did microcosm studies, we found that with ethane added, the organisms that were growing in the aquifer, and we isolated an organism from that aquifer that grew on ethane and degraded 1,2-dibromoethane. Although the gas is slightly more expensive than propane, the kinetics were better, so the rates of degradation were better with ethane, and the concentration achievable based upon some of the studies we did, with ethane, we saw that we could get down to the detection limit, which was 15 parts per trillion, which was well below the federal MCL, and also which is 50 parts per trillion, the Massachusetts MCL, which was 20 parts per trillion. And Mike Hyman is doing some work with his grad students with some of those organisms, and there do seem to be cases where ethane either induces some enzymes more strongly or induces slightly different enzymes, or just in there again, a lot of this comes down to which enzymes are being induced and sort of the kinetics of degradation of your contaminant of concern. And so ethane, in some instances may have some select advantages over propane.
Okay, next question, Paul. Is it common to find competitors for substrates that aren't actually performing the cometabolism? And if so, what would be the best strategy to ensure that you're getting cometabolism of your contaminant of concern?
That's a great question. So the one thing that's fortuitous, let's put it this way, is that a lot of these different organisms, and many of them fall in this group, this order Actinomycialces, or they're actinomycete type organisms. There seems to be a lot of conservation of these enzymes. So if you looked at the data that I sort of showed you from that project where we did the bioreactor, the organism we added, ENV 425 Rhodococcus, was not there later on in the demonstration, and yet the system still worked extremely well. So in that case, the organism that we added probably just didn't do terribly well in the Las Cruces, New Mexico groundwater, where there were natives that did extremely well. So they displaced our organism in the reactor, but the system continued to work great. So because there's some conservation of these enzymes, the good news is that for a lot of these substrates, again, things like propane, ethane, methane, the alkane gases, there's sort of a limited suite of enzymes that break these down, and many of them have cometabolic activity. Now, that being said, some of Mike Hyman's recent work with Lou Semprini has shown that there are multiple enzymes that are induced with propane, and some of those enzymes, the traditional propane monooxygenase, actually doesn't degrade many of these contaminants. In fact, it's a second oxygenase, which Mike has termed short chain alkane monooxygenase. There's different names for it, but that's the enzyme that's actually performing most of the degradation. But oddly, in all but a few instances, these bacteria contain both enzymes. So if you add propane, you're inducing both the propane monooxygenase, which does nothing, but you're also inducing the short chain alkane monooxygenase, or SCAM enzyme, which degrades most of these contaminants. Now, in order to hedge your bets, and in some work that we've been doing more recently with Lou and Mike and Tony Danko, where I'll do a lot of co-metabolic work, isobutane only induces this SCAM enzyme. So by using isobutane, you're sort of hedging your bets that you're inducing the correct enzyme. But I've not seen an instance where we've added propane for a variety of different contaminants, and when propanotrophs have come up, so when the propane gets degraded, I can't remember one instance where the contaminant we were looking at, and it was typically either 1,4-dioxane or NDMA or some of the CVOCs didn't biodegrade. So I think that's at least for the alkane gases kind of an unusual, a rare circumstance. I don't want to speak as much for things like toluene or phenol, because I don't have as much background with those in terms of the different enzymes that break them down. But that's a really good question.
Yeah, another question that we've gotten a couple of versions of, and I think people relate to, is, we talk a lot about reductive dechlorination and these anaerobic conditions. So how would you engineer aerobic cometabolism to be down gradient of these anaerobic treatment zones?
Yeah, another really good question, and so yeah, you do have these plumes where you reductively treat the source area, and then perhaps you get some vinyl chloride or cis DCE, or some of your parent compound moving down gradient. So there's a couple of ways that you could do this. Again, a lot of this also depends on the geology, right? It's if you have a shallow aquifer that's very conductive, a lot of this is easy. If you have an aquifer that's 500 feet to groundwater, or it's in fractured bedrock, a lot of this is hard. So I'll speak to the easy case, which is, geez, you have a shallow plume, it's 10 feet to water, it's in a sandy aquifer in New Jersey. So what you can do is you could start by just putting in a system where you add air. So you do your anaerobic treatment up gradient, and then down gradient, you have a system where you try and recreate aerobic conditions to see if, in fact, some of those substrates that are coming down, let's say you've added an emulsified oil up gradient, you've gotten, most of the degradation of your chlorinated solvents, but you still have 1,4-dioxane moving down gradient. So one option would be to put in sort of an air sparge barrier where you're adding oxygen and reoxygenating and seeing if, in fact, there's enough carbon source coming down that you can promote aerobic degradation of 1,4-dioxane. Or you put in a system where you can add both oxygen. You're going to probably have to add inorganic nutrients there because a lot of those will have been consumed up in the source area. So where you can add diammonium phosphate or ammonia gas, and a carbon source, something like propane or isobutane or ethane. And by that, you're sort of creating these conditions where now you're becoming reoxidizing again, and you're adding another co-metabolic substrate and some nutrients so that you can promote that sort of final degradation of those contaminants.
Okay, so this next comment is really cute, Paul. I think you'll like it. It says, great presentation. Thank you for banging the co-metabolism drum. The question that he has is, have you seen any issues with the re-injection in circulation cells, such as biomass or abiotic product formation impeding the flow of these systems?
Yes, yeah, I mean, that can be an issue. It's like any injection system. So if you sort of get to a point where you're going to run particularly a research system. So, if you're running a sparging system, a biosparging system, and you're doing it properly, you typically are not going to have issues with sparging. And when I say doing it properly, I mean, you're sort of periodically adding gases. You're not moving any groundwater in that case. So you typically have less issues there. But that being said, if you're shutting those wells down periodically, it's never a bad idea to add something to keep biomass from growing. But if you're sparging, we've run these systems for multiple years with the sparging systems and typically have not had issues with the pure sparging systems. Now, when you get into any type of research system where you're pumping groundwater, biofouling can always be an issue, and you really have to sort of design in approaches for that. And the best way to do this is you recirculate periodically, so that you're just using the recirculation to kind of mix your amendments, if you can do that. If you can't do that, if you have to recirc, continually, kind of like we did at Cape Cod, then you've got to think about adding amendments periodically or redeveloping your, particularly your injection well, because that's where you're probably going to see, biomass formation is in your injection well. So things like hydrogen peroxide, acid, citric acid, there's different ways that you can do that, but you're probably just going to have to think about periodic maintenance of that injection well or that series of injection wells if you're doing recirculation, but again, much less so if you're doing biosparging.
Okay, two combined questions here. Are you using commercial labs to carry out the analysis that you're doing? And can you also talk a little bit about the well spacing, how you would recommend for sample collection?
Yes. So yes, this is Microbial Insights, and we've worked with them for a lot of years because they do a great job at this. So they've got a lot of tools for the qPCR for aerobic cometabolic organisms, as well as offer that C14 assay. So those are two things that are done through MI. They also have this large database that as clients you can access and sort of take a look at overall findings. Well spacing is highly variable. I used to kid with Dave Lippincott a lot that we do all this modeling every time we do one of these sites, and it seemed like the well spacing was always 15 feet, at the end of it. But no, it can be highly variable based upon conditions, and normally, so if we're doing a biosparging system, the first thing we do is we put in one sparge well, and then we put in a series of monitoring wells, or use existing wells and go out and do tests where we sparge in a certain amount of gas, and typically these days we just use air, and then look for changes in DO in existing wells and changes in mounding. So that's really kind of what you should do, and that way that'll tell you what your distribution is for your gases, and that that becomes pretty important whether you're designing a barrier, or you're designing a a different type of treatment system. But that's kind of the approach. My default would always be 15 feet, but that's not true. There are cases where we've added gases and seen influence 50 feet away or more. So it depends on the formation. Are you in a confined interval? Are you in an unconfined zone? How big is that interval? And again, for the recirculation, so at this project we did up at Cape Cod, that those wells were the injection extraction well were about 60 feet apart, and it's kind of how long you can sort of run the flow of water. But I think we could have done that even over a much larger area. But that's really an engineering geology question in terms of getting your amendments distributed in the aquifer.
Okay, I have three questions that relate to substrates. I'll kind of bulk them all together for you to talk about. The first one is, have you used calcium peroxide? And then the second one is alcohols being used as a substrate. And then the third question is, why did you choose DAP compared to MAP for the phosphorus nutrient?
Okay, so calcium peroxide. I don't know, in terms of that wouldn't act as a co-metabolic substrate. Calcium peroxide, maybe the question has to do with using it as a well treatment to basically to prevent fouling, but I don't have a lot of experience with that one. I'm sorry, Dora, what was the second one? Oh, alcohols. Yeah, so that's a good question. The primary, so for a lot of these oxygenase enzymes, the primary product they create is an alcohol. So methane goes to methanol, ethane goes to ethanol. That's sort of the first reaction. So one of the things that folks have been trying to do, and us included, is to figure out ways that in some instances you could increase biomass further or increase biomass without inducing the oxygenase. I know that seems a little complicated, but we ran a reactor project recently and we were trying to grow biomass quickly, and we were using propane. So we fed some 1-propanol into the reactor, and the organism grew very well, and it attached to the biomass. So it was a way to really stimulate growth of the organism over a short period of time. Now, that being said, there's a risk with the alcohols that you're going to basically grow organisms that don't have the oxygenase. So that's the one potential downside of that, is that you might add propanol to an aquifer if thinking that, okay, I'm going to stimulate a lot of co-metabolic treatment here. But if the organism that grows up doesn't have the oxygenase, that's probably not going to happen. And even if the organism that grows up does have the oxygenase, the propanol may not induce it. So you may be growing the organism but not seeing degradation. Then if you turned around and added propane gas and you had a good population of the organism that had the oxygenase, then it might really take off. But the risk is you're not growing the right organism. So I'd answered that question earlier just with the gases, but with the alcohols, that could be a potential problem. Diammonium phosphate rather than, I think, monoammonium, I think, was the question. And some of it's more bang for your buck, I guess. You get 2 ammonia molecules per mole instead of 1, and it's really what you're trying to add. I mean, the phosphorus, I haven't seen many instances where phosphorus seems to be limiting. It's often nitrogen that's limiting. So that's sort of part of the reason for that. It's also cheap and readily available.
I have another 2-part question for you here. If you had to choose between doing a bench test and a C14 study, which would you pick? And then if you do a bench test, are there certain co-substrates that you would recommend that should be evaluated?
Yeah, I guess part of that is what is your contaminant profile, and sort of what are you trying to show? So I see the C14 test having the most applicability if you are trying to understand whether there's natural attenuation going on of a chlorinated solvent. So if you're trying to show natural attenuation, taking a sample from the field and immediately running this C14 study is going to give you probably the best evidence of whether something is actually occurring at the site in that location. Now, in terms of a traditional test, we often do those, and part of that is to determine if you can promote activity. So if you're going to go into the field and you plan on doing something, you plan on adding a co-metabolic gas or doing a sparge system or a recirc system, and you want to see if 2 things: A, which of your substrates is the most effective, and then B, are the organisms there that can degrade those substrates? So often what we'll do is, we'll start microcosms. We'll add several different substrates individually, and we'll add things like methane. Often we choose, in terms of the substrates, methane, ethane, propane, isobutane, just for the contaminants we're interested in. And then what you look for first is, are those gases being biodegraded and at what rate? And if the populations are growing, you add those to bottles and they get consumed. You add them, they get consumed. And then often what we do, we'll do that for a month or 2. And then when we know we have a good population, we'll spike in our contaminant of concern. And then we'll take a look at sort of which population most effectively biodegraded the contaminant we're interested in, be it 1,4-dioxane or chlorinated solvents or NDMA. So in that way, you're saying, okay, if you're trying to show natural attenuation, this is kind of what I'm going to be doing in the field. I'm going to be adding a substrate, at certain intervals, and I want to see what happens in a bottle test. And I'll choose the substrate in part based upon, which of those microcosms has the best kinetics for degradation of the contaminant of concern. And then there's secondary factors as well when you're choosing a substrate. Certainly cost is one of them, but solubility, vapor pressure can make a difference. Things like, propane and methane have a high vapor pressure. You get up to things like isobutane, they have somewhat lower vapor pressure. So you kind of have to think about that when designing systems. But in choosing a substrate, its efficacy, I think, is number one. Gas characteristics are probably number two. And then, you can look at cost, but again, in most instances, the cost of the gas is not a particular driver for this technology.
All right, one last question, Paul, and we'll let you go. I appreciate you staying over with us. The question is, did I hear correctly that aerobic cometabolism will occur under depressed pH, down gradient of enhanced reductive dechlorination?
Yes, so I currently have a CertUp project with Bella Chu at Texas A&M and David Friedman at Clemson. And what we've been doing is trying to understand whether you can stimulate degradation, aerobic cometabolism of chlorinated solvents at low pH. So, pH below 5.5. And we've had significant success doing that. We're looking at methane in particular as a substrate, but also looking a little bit at ethane and propane as well. But we've been collecting samples from low pH sites, and if someone who's still on the phone has a low pH site that they're interested in, please contact me. My contact information is on the screen, because we're still looking for one more site, or potentially others to sort of do this work. So that would be kind of some free work at the site. But yeah, so we've seen degradation at pH values down into the 3s. So it's certainly a potential, and it's not something you're going to typically see reductively.
That's exciting. Well, thank you very much, Paul, for staying over. I know you're 30 minutes extra, so I appreciate you doing that for us. And thank you, everyone, for hanging on and listening. If you have more questions that come up, feel free to reach out to Paul. He's a great resource for us and so much experience in this area. His information is there. You can also always email us at info@microbe.com. and we will be happy to get with Paul or to give you any information that we have. But again, we appreciate, Paul, you taking time and this excellent presentation and sharing your wealth of knowledge with us. A lot of thank yous coming from the audience right now. And we thank you, everyone, for hanging in and listening on the webinar. And hopefully you will join us for future webinars that we host. Thank you again, and thank you, Paul. Have a wonderful day.
Great. Thanks, everybody. Thanks for taking so much time. Thanks, Dora.
Thanks, Paul.